In section 5, we are talking about how messenger RNA goes into becoming a protein. So in this section, we're going to be talking about what each one of the RNAs does in making a protein. So translation can also be referred to as protein synthesis because it's going to be making a protein. So translation converts mRNA message into polypeptides. Polypeptides are going to be a chain of amino acids that are joined with one another, and the polypeptides will join, those polypeptides will join up with one another to make a protein. So when we are reading messenger RNA, we are looking for a codon, which is a sequence of 3 nucleotides that code for an amino acid. So down below is a segment of messenger RNA, and we split those into groups of 3. So they've already sort of split it. I'm just going to split it a little bit more. So you'd read 1, 2, 3. That's going to be an amino acid, and that first one is methionine. The next one is leucine. So those nucleotides, those group of 3 nucleotides, are called a codon, and they code for an amino acid. Most of those codons are going to code for a specific amino acid. Some of them don't code for an amino acid at all, and we'll talk about those here in a minute. But the methionine, the AUG, is the start codon of all proteins. You'll end up finding that one being the start of it. Genetic code matches each codon to its amino acid or function. There are 3 stop codons, 1 start codon, and it codes for methionine. So as you look at this table over here on the page that just showed up, I'll explain how you read that. So first, if we're looking for, let's say, the AUU, for example, we would end up saying, okay, the first base, again, if we're going A, U, U, we would go, and the first base is going to be this column over here. So we look and we go, okay, here's A, so we know it's going to be somewhere in this row here. Okay, then it says find the second base in the row and look at the top, and let's say we are looking at the U. So we go up here and we see right here that we've got the U. So we go down, we find that, oh, it's going to be somewhere in this box here is our amino acid. And then we look and we see U was our final one, so we come over to the third base and we follow that all the way across, and we see, oh, it would be isoleucine would be the base that AU, or the amino acid that AUU would code for. So that's how you use this chart. So again, the final one, you come over and here's the third base. So if I end up having you do that on an assignment or things, you'd be able to figure that out. So that's how that chart's read. There's ones that's also a circle, and it's the same thing. You just use the concentric circles going in. A change in the order in which the codons are read changes what the protein that's being made. So every protein has a specific order for its amino acids. And then you also have a specific arrangement and sort of structure of those amino acids on how they bend and twist and things like that. So a change in the order in the codons, again, will be different. So you can look from reading frame 1 to reading frame 2. You can see that the reading frames are messed up, and so you're getting different amino acids being created. We'll talk more about this when we talk about the mutations that can happen. So this right here would be what's called a frame shift or a reading frame mutation. So regardless of the organism, codons code for the same amino acids. Amino acids, like I said, are going to link together to become a protein. When we are bringing amino acids in, there's going to be something called an anticodon. The anticodon is on the transfer RNA. So the anticodon is a set of 3 nucleotides that is complementary or opposite. Anti is against. So it's opposite of the messenger RNA codon. The anticodon, like I said, is carried by the tRNA, and that's what you see pictured there. And right here is the anticodon. It's showing that there. And on the end of it, the transfer RNA is the amino acid that would be for whatever that anticodon is. Another part that's going to play a major role, if you remember back to when we were talking about the structure and function of cells, was the ribosome. The ribosome's job is to make proteins. So the ribosome is made up of ribosomal RNA and proteins, and it's going to be 2 subunits. And there will be a picture that will come up here in a second. So the large subunit has 3 binding sites for the transfer RNA. So you're going to have 3 transfer RNAs that are going to land in the binding site area. And then you're going to have small subunit of the RNA of the ribosome that's going to hold the messenger RNA. So this whole thing, this little green thing, would be sitting on a messenger RNA and going along. So for translation to begin, transfer RNA is going to bind to the start codon and signal the ribosome to start assembling. A complementary transfer RNA molecule binds to the exposed codon, brings its amino acid close to the first amino acid. So again, really the first thing that's going to happen is the messenger RNA is going to leave the nucleus. And when it leaves, the ribosomal RNA is going to basically sit down on that messenger RNA and read it. And when it reads it, it's going to signal we need this amino acid. So the transfer RNA is going to bring it into this first spot here. And that's what's right here. That first one had landed there, and it had an amino acid. And then the next one comes in and lands, and when they land, the 2 transfer, so this would be the second one that came in, the 2 transfer RNA are going to make contact with one another, and the oldest one is going to leave its amino acid behind with the newest one. And then you'd have the third one come in and land right there, and then those would make contact and the whole thing would shift down. When it shifts down, that transfer RNA that was 1 leaves and is able to go pick up another amino acid. And it's just going to keep going. As you see in this picture over here, you can see that that polypeptide is being made, and it's getting even longer here, and you can see that it's twisting and turning. So that is the process of making a protein. So again, the job of the messenger RNA carries the code on how to make the protein. The ribosomal RNA is going to help sort of read the messenger RNA and signal what transfer RNA needs to come in, and then the transfer RNA is going to bring the right amino acid to the ribosome. So the ribosome helps form a polypeptide bond with the amino acids. The ribosome pulls the messenger RNA strand the length of 1 codon. It's going to pull it along, just like I was saying before. So that exposed codon, it's going to get pulled in, and you're going to end up having the transfer RNAs coming in there and landing and making contact. The now empty transfer RNA molecule exits the ribosome, and a complementary tRNA molecule binds to the next exposed codon. Once the stop codon is reached, the ribosome releases the protein and disassembles. So you can see right there, it gets to the last one. There's nothing that codes for that. So the ribosome's like, and we're done. Well, what will usually happen is that protein or that polypeptide that's made will go be secreted into the ER, the rough ER, and it will be processed more. It will probably be connected with other polypeptides to officially make the protein. So, and then once it's all made, it will be shipped out by the Golgi apparatus. If you guys remember, those were the three things that were involved in making a protein within the cell. So here was the actual process of protein synthesis. So that's the end of the notes for section 5.